Hello everyone, welcome to IS Baba's 60 days rapid revision series for Prelims 2022. This is date 35 and we take up the science and tech here. So virology and then pathology. So these are the topics of discussion. Now coming to the first one, the TRIPS policy relaxation. So here, when the COVID-19 pandemic broke out, so the developing countries like India, South Africa and others, they asked the WTO for the relaxation. So what was the relaxation? It was the compulsory licensing. That means immaterial of whether a drug is having patent or not, if a compulsory licensing is issued on that drug, so that means any company they can produce that drug without providing any patent money for the owner. So that compulsory licensing is provided under the Doha Declaration for WTO and the Article 30 of TRIPS. So that provides for the TRIPS flexibilities. That is during emergencies. So we can go for the flexibilities or relaxations of these TRIPS. And here, it was claimed by the developing nations like India, South Africa and others. And the proposed relaxation says that the compulsory licensing will be granted to those nations whose pharma market is less than 10% of the world market. So here, the pharma markets like India should not misuse this compulsory licensing. So that is why the WTO stipulated that only if our market is less than 10%, we can produce this drug. So that means if our market is less, so we are using this compulsory licensing only for the production of the drugs to our nation and only to wait away the pandemic. We are not making the commercial use out of it. So commercial use means I am not producing en masse drug and I am not making money out of this situation. So this is all about the TRIPS relaxation. Then One Health. So friends, we all know that One Health means we are clubbing the human health, animal health, insect health, soil health and all others. So go with the inclusive approach. So all pathogens, so they are clubbed under one domain in this One Health concept. And the Department of Biotechnology in the Ministry of Science and Technology supported a mega consortium on the One Health and launched the first One Health project of DBT. And this consortium consisted of 27 organizations led by DBT's National Institute of Animal Biotechnology. So these are the nodal agencies for conducting this consortium. Then this program envisages carrying out surveillance of important bacterial, viral and parasitic infections of zoonotic as well as transboundary pathogens in India. And then use of existing diagnostic tests. Then development of additional methodologies when required are mandated for the surveillance. So the research and development in all those fields and the interdisciplinary fields. So these are the core concepts of One Health. And then the origin of One Health. So the father of modern pathology, that is Rudolf Virchow. So he emphasized in 1856 that there are no dividing lines between animal and human medicine. So from here the word originated. Then One Health vision derives its blueprint from the agreement comprising the FAO, that is the Food and Agriculture Organization of United Nations, and the World Organization for Animal Health. So these two organizations, they were instrumental in bringing this One Health vision. Then the overarching purpose is to encourage collaborations in research and sharing of knowledge across various disciplines, that is animal health, plant health, soil health, then human health, all others. The term One World, One Health in 2007 along with its 12 recommendations. So the 12 recommendations were also called as Manhattan Principles or Manhattan Principles. So these focused on establishing a more holistic prospect to prevent epidemic diseases. So here, make sure that this Wildlife Conservation Society, so this originated the word One World, One Health and its Manhattan Principles, which stated that an inclusive approach for preventing the pandemic outbreaks. And these are some of the facts. Friends, for sustainable development, so many facts were asked by UPSC. So on those lines, I have brought some of the facts for this One Health. So it is better to remember because these words, it was in news many times. Then come to next, the lymphatic filariasis or elephantiasis. So recently Maharashtra government has started a drug administration drive for elimination of lymphatic filariasis. And here, what is this lymphatic filariasis? That is LF, commonly known as the elephantiasis, and is considered as the neglected tropical diseases. Friends, there are many tropical diseases that happens in the third world and they are neglected because no research and development goes on for inventing drugs and vaccines for them. So that is why they are put under the neglected tropical diseases. Then it is the second most disabling disease after the mental health. So mental health is the first and filariasis is the second in disability or disabling capability. Then it impairs the lymphatic system and can lead to the abnormal enlargement of body parts causing pain, severe disability and social stigma. So elephantiasis, the legs and the parts that is infected that will swell like anything, and that is the meaning of elephantiasis. So how this swelling occurs? That is mainly due to the damage of the lymphatic system. So we know that along with our circulatory system, we have the lymph nodes, lymph vessels and the lymph fluid. And now if a worm, if it attacks the lymph node or a lymph cell or a lymph fluid, so it blocks the lymph that is flowing in that part. So if this is the leg and here if it is a lymph vessel, so it blocks the lymph and all the lymph that comes from top, so it will accumulate. That leads to the swelling of the legs. Then lymphatic filariasis is a vector-borne disease caused by infection with the parasites classified as the nematodes or the roundworms. So we can also call they belong to the phylum Aschelminthis and they are belonging to the family Filariidae. So remember the Filariidae, the family and the class nematode and the phylum Aschelminthis. Then some of the species Wuchereria bancrofti. So it is responsible for 90% of the cases. Then Brugia malayi causes most of the remainder of the cases. So malayi means Malaysia. Then Brugia timori or the Timor. So this also causes filariasis. So these are some of the facts. Then come to next. The WHO recommends 3 drug treatments to accelerate the global elimination of lymphatic. So what are the drugs? The drug treatment known as IDA. So that involves combination of ivermectin, then diethyl carbamazine citrate and albendazole. So that is I, D and A. So remember ivermectin and diethyl carbamazine citrate and albendazole. So also remember this albendazole. It is also used as the deworming tablet. So in midday meal scheme along with that, so every year we give this albendazole so that if there are any worms in the intestine of the children, so that will be dewormed. Then the global program to eliminate lymphatic filariasis, that is the GPELF program. So in 2000, WHO established the GPELF to stop transmission of infection with mass drug administration and to alleviate suffering among the people infected. So by morbidity management and disability prevention. So here treatment and then the relief for the symptoms and then care. So all these, so these are being taken up under this GPELF. So remember the medicine and also the program. Then come to next, the visceral Leishmaniasis or the Kala Azar. So here recently a team of researchers from the National Center for Cell Sciences, Pune, have found new biomolecules to fight drug resistance in Kala Azar. Friends, this Kala Azar, it is again a deadly disease in Bihar, Jharkhand region, and that's why it becomes important. And here, the National Center for Cell Sciences is an autonomous organization under the Department of Biotechnology. That means Ministry of Science. Then Leishmaniasis, it is a neglected tropical disease, again just like the filariasis, and it is affecting almost 100 countries including India. Then it is caused by a parasite called Leishmania, which is transmitted through the bite of sand flies. So remember this Leishmania is a bacteria, it is not a worm, and this is transmitted by the sand flies. So this is a vector, sand fly, and Leishmania is a family of bacteria. Then come to next, there are 3 types of Leishmaniasis. One is the visceral Leishmaniasis, which affects multiple organs and is most serious form of disease. And then cutaneous Leishmaniasis, which causes skin sores. So it is also most common. So here cutaneous Leishmaniasis means you will get rashes. So once the fever comes and the fever is cured, so you will be having rashes and that remains for 2 to 4 months. And then we have mucocutaneous Leishmaniasis. So that means mucus plus cutaneous. So here the fluids like the sputum, then the mucus and also the skin. So which causes skin and mucosal lesions. So these are the 3 types, which is multiple organs and which affects only the skin and which affects both the skin and the mucus. And then the visceral Leishmaniasis, which is commonly known as Kala Azar in India, is fatal in over 95% of cases if left untreated. And then the only drug available against Leishmaniasis is the miltefosine, and it is rapidly losing its effectiveness because of emerging resistance to this antibiotic. So here the antimicrobial resistance is playing and miltefosine, so this is losing its effectiveness. So this is all about the visceral Leishmaniasis. So basically the symptoms are the fevers, and after fevers we also get the organ damages, or we can get the rashes, or we can get the sinusitis due to blockage of mucus and fluids and others. So this is all about the Kala Azar. And then come to next, the Mucormycosis and the Candidiasis. So we know that the black fungus and white fungus diseases, so the same which happened after the COVID-19 pandemic we are discussing. So Mucormycosis is a rare but serious infection that is caused by a group of molds called the Mucormycetes. And this is a fungal disease and it is termed as the black fungus, and it affects people who have health problems or use medicines that depress the body's ability to combat the germs and illness. So who are having depressed immunity, so their immunity is being suppressed due to some medicines, and those people are most vulnerable for this mucormycosis, and it reduces the ability to fight environmental pathogens. So whatever pathogens they are around us, even that we can't fight. In that stage, these mucormycosis and candidiasis will infect. Then it most commonly afflicts the sinuses or the lungs after inhaling fungal spores. So it affects the lungs and also the sinuses. Friends, what is a sinus? It is the nasal cavity. So this nasal cavity is present in the interior part of our forehead. So between our eyebrows, so that is the exact part of sinuses. Say for example, sinusitis, it is a disease. So that means the mucus is stored in that sinus, and once you bend your head down, it pains near the eyebrows. So the space between your eyebrows, it pains. So this is the sinuses, and most of these black fungal diseases, we knew that it started from the sinuses and then it spread into the eyes, then cheeks, and the whole face become blackened and the person died. Then it can also harm on the skin after a burn or cut or other type of skin wound through which the fungus enters the skin. So any way, so the fungus can enter. It need not be one particular way. Then people having comorbidities, that is the treatment for varicose veins and others, and then uncontrolled diabetes mellitus, and then immunosuppression by steroids and prolonged ICU stay. So these are the victims of the mucormycosis. Then come to next. So here we have the rhinocerebral mucormycosis. That means it affects the sinus and the brain. And then pulmonary, so it affects the lung. And gastrointestinal, so it affects the digestive system. And then the cutaneous, that means it affects the skin. And then disseminated mucormycosis, so it enters the blood and it can affect any part of the body. So these are some of the types of fungi. And then come to next, candidiasis or the white fungus is a fungal infection caused by yeast. Okay, so it is a fungus called candida. So candida normally lives on the skin and inside the body in places such as mouth, throat, gut, vagina without causing any problems. But candida can cause infections if it grows out of control or if it enters deep into the body. For example, bloodstream or internal organs like kidney, heart or brain. So external organs they can tolerate, but internal ones they can't tolerate. Then the most common species that causes infection is the Candida albicans. So remember Candida albicans and the white fungi, the collective name or the common name. Then the XE and XF variants of COVID-19. So recombinations occur when two different lineages of virus co-infect the same cell in the host and exchange the fragments of their individual genomics. So this XE and XF, we got to know they are the recombinant variants. So how these recombinant variants are emerged? Say for example, if two variants, so they are simultaneously infecting a person. So these two variants, say for example, the Delta and Omicron, they infected the same person at a time. And now they will exchange their gene content and now the mutation occurs. And this is how the recombinant variant is born. And here the difference between the recombinant variant and the mutation variant is, the mutation means either addition or deletion of the RNA takes place within one cell. But here in the recombination, the exchange of genetic material takes place between two variants. So these recombinant lineages are annotated by Pango with an X followed by an alphabet which indicates the order of discovery. So Pango is a network, so that is giving the name for these recombinant lineages, and this is also embedded with a software which will identify various lineages. So we discuss about Pango for a while, and as of now we discuss these X variants. So XA, so it is a recombinant of the B117 alpha variant and B177, and this is detected in UK. And then we have the lineage XB that is detected in US, and then the lineage XC detected in Japan, and which is a recombination of B117 the same and a delta lineage AY29. So just try remembering these, you need not by heart. Then the three new recombinant lineages designated by Pango network are being monitored as the XD, XE and XF. So here the lineages XD and XF, they are the combination of lineages AY4. Again, the sublineage of Delta variant and BA1, a Omicron. So the exchange of gene took place between a Delta variant and a Omicron variant. So crossbreed between the COVID second wave and the third wave. Then XE is a combination of Omicron lineages between BA1 and BA2. So this XE is the recombination of the Omicron variants only. Then the lineage XD was first detected in France and Denmark, then XF in UK, and then XE first detected in UK again. And then Pango network. So it is an open global consortium of researchers from across the world and provides a system for naming the different lineages of the SARS-CoV-2. And the Pango means the phylogenetic assignment of named global outbreak lineages, or the Pango lineages, or the Pangolin is a software tool developed by the members of the laboratory of Andrew Rambot. So remember, the Pango is a network and Pangolin is a software. So these are some of the facts. Then come to next, WHO's Solidarity Clinical Trial for the COVID-19. So here India is all set to join the WHO's Solidarity Trial, which aims at the rapid global research for the drugs to treat COVID-19. So here the Solidarity Trial is going on in the 4 drugs. That means the first, the remdesivir is being tested for its ability to cure COVID, and then the combination of lopinavir and ritonavir. So these are the anti-HIV drugs, and their effectiveness is being tested on COVID-19. And then we have interferon beta, so that in combination with the lopinavir and the ritonavir. And then the chloroquine, so one of the TB drug, it is also being tested. And here these drugs are tested for their effectiveness to the standard of care and also the regular support. So that means with the minimum care and support at the hospitals, so these drugs, they should cure COVID. So minimum efforts, maximum gain. That is the aim of the study. And then the Solidarity Trial was initiated by WHO and its partners, and it is the world's largest multinational human trials on COVID-19 therapies. So remember the term largest here. Then the trial covers 4 repurposed drugs. So the same remdesivir, hydroxychloroquine, and then leponavir, ritonavir, then interferon. So these are the drugs, and India has entered the research recently. So these are some of the facts. Then come to next, 2-deoxy-D-glucose, that is 2DG. So this 2-deoxy-D-glucose or the 2DG is an anti-COVID oral drug. So an oral drug, very easy to administer, and it has been developed by the Indian Institute of Nuclear Medicine and Allied Sciences, the INMAS, and it is a laboratory of DRDO in collaboration with Dr. Reddy's laboratory. So remember, this INMAS is a lab of DRDO. So a skewed thing, that is defense, coming into the medical sciences. Then the 2DG comes in powder form in the sachet, and it is taken orally by dissolving the powder in water, and the drug accumulates in the virus infected cells. So this is important for us. So how it will treat? It will go and accumulate in the virus infected cells, and then thereby it prevents the virus growth by stopping viral synthesis and energy production. So remember, the synthesis of viruses or the reproduction and the energy production is being impacted. Then the selective accumulation of the drug in virally infected cells makes this drug unique. So it accumulates only in those cells which are being infected by the virus, and it will not accumulate in any other cells. So that is how it is effective. So this is all about the 2DG drug. Then the drug helps in the faster recovery of hospitalized patients, and further it also reduces the supplemental oxygen dependence. Then a higher proportion of patients treated with 2DG also showed the RT-PCR negative conversions. So that means even in the RT-PCR, we got the negative report after treating a patient with this 2DG drug. So that means it is highly effective. Then the DCGI, that is the Drugs Controller General of India, has approved this drug for the emergency use. So approval has also been given. So this is the 2DG. So remember the oral drug. Then description of the Covishield, Pfizer and Sputnik. So we take first one, Covishield. Covishield has been prepared using the viral vector platform. So that is in the vaccine, a chimpanzee adenovirus, that is CHADOX1. So this has been modified to enable it to carry the COVID-19 spike protein into the cells of humans. So this chimpanzee adenovirus is the vector, and within that the spike protein is being sent into the body. And this cold virus is basically incapable of infecting the receiver. So this virus is not virulent, so only the spike protein, so that is exposed. And once the spike protein is seen, the body triggers its immunity. So this is how Covishield works. Then come to next, the Sputnik 5. So Sputnik 5, the world's first registered vaccine. So mark this as important. It is based on the human adenoviral vector based platform. So it is not the chimpanzee vector. Now it is the human adenovirus. And the vaccine uses dissimilar disarmed strains of adenovirus that cause the common cold. And these are used as vectors to deliver the jab. So here it is like the one dose of Sputnik that uses one strain of adenovirus, and the second uses the second strain of adenovirus. So here there are least chance of this adenovirus they infecting the cells. So that is why this Sputnik was considered more safe than the Covishield, and it was costly as well. Then come to next, Moderna's mRNA 1273 vaccine. So a vaccine based on the messenger RNA. So that is the mRNA technology. So we have discussed what is mRNA. So this Moderna is an mRNA vaccine. Then Pfizer. So this vaccine is composed of the nucleoside modified mRNA. So here we have a mRNA or a modified RNA. So this RNA codes for the spike protein. So again this Pfizer, it is also a mRNA vaccine. So this is also worth remembering here. Then come to next, covaxin. So India's first indigenous vaccine against COVID-19, and this covaxin is in fact an inactivated viral vaccine. So this is not using any mRNA technology. It is inactivated the COVID-19 virus only. Then the vaccine is developed with the whole virion inactivated virus cell derived technology. So anyhow, this is a technology to extract the inactivated virus, and this covaxin, so it has some chances of becoming revirulence. So it is an inactivated COVID-19 virus only. And that is why there was some skepticism that grew among the public, and that is why there was some initial aversions to take covaxin. But anyhow, now we have most of our population being vaccinated, so which is a welcome step. Then the covovax, so or the Novovax. So this uses baculovirus to produce COVID-19 related antigen. So here the baculovirus is used, and it is developed by the CEPI. So remember this. And then the covovax, it is an inactivated virus based COVID-19 vaccine developed by the Russian Academy of Sciences. So covovax is very similar to covaxin. But however, this covovax is different. So here this baculovirus is modified in such a way that this will produce a COVID-19 type antigen. So here some genetic modification of baculovirus, so that takes place. Then come to next, then corbevax. Friends, here the difference is that corbevax uses this yeast called Pichia pastoris. So instead of any virus or any other bacteria, so we are using Pichia pastoris, a yeast, as a vector, and this yeast, so that will produce the spike protein when it is injected into the cell. So remember the Pichia pastoris and the fungus here. Then come to next, the spot, that is the scalable and portable testing. So this is one of the diagnostic tools of the COVID-19. So how to diagnose? The spot gives COVID-19 test results in 30 minutes. So very fast, and the device can be operated by anyone with minimal training. So very easy to operate as well. Then the test is very much similar to that of RT-PCR. That is the reverse transcription and polymerase chain reaction, the same procedure goes. But it uses reverse transcription loop mediated isothermal amplification, that is the RP-LAMP technique. And however, it does not need complex machinery or expertise. So this RT-LAMP technique is different from the RT-PCR. So we don't require an RT-PCR machine per se to do this RT-LAMP technique. So how to do this? This RT-LAMP technique is one-step nucleic acid amplification method. It is used to multiply the specific sequence of RNA. So here, when we discussed about RT-PCR technique, we found that we are developing an entire sequence of COVID-19 RNA, and this synthetic RNA, so that will go and bind to the actual COVID RNA. And now the DNA is formed with a reverse transcription. But here, this much of work is not required. So we can take one piece of RNA, and then we can bind it with the other piece of actual COVID-19 RNA, and this one piece, so that can detect the positivity of the COVID. So as the effort to synthesize the whole sequence is reduced, so this is much simpler. That is the RT-LAMP is much simpler compared to the RT-PCR. So remember, the only difference is here a segment of RNA is used, and in RT-PCR the whole chain is being produced. Then come to next, the National Anti-Doping Bill 2021. So the bill seeks to provide a statutory framework for functioning of the National Anti-Doping Agency. So earlier it was not a statutory body, it was an executive body, and now the statutory status is being given. And it also empowers this body to conduct raids and investigate and then levy sanctions, or to set up anti-dumping rules in case of violations. So more powers are being given. Then the bill provides for the establishment of the National Dope Testing Laboratory. So one centralized laboratory for the testing of various doped people. Then the bill says that the accounts and the audit of the board of NADA and NDTL will be done by the CAG. So all auditions will be done by the CAG himself. So that means a kind of credibility given. Then the bill lays down the circumstances, acts or conducts which will constitute an anti-doping violation. So which and all acts will violate the rules, so that will be mentioned in detail. So here a detailed policy is being enacted, so again it becomes important. Then the bill also seeks to establish a national board for the anti-doping in sports. So this national board, it is a regulatory body, and this will consist of a chairperson and 2 other members to be appointed by the central government. Then this board will constitute a national anti-doping disciplinary panel and the anti-doping appeal panel for the purpose of determining the consequences of the anti-doping rule violations. So here the disciplinary panel will take up the disciplinary action, and the appeal panel, so that will take up the appeals from the disciplinary panels. So these are some of the administrative and the technological bodies being established by the anti-doping bill. So we have to remember all these. Then come to next, the countdown to 2023. So what is this countdown to 2023? In 2018, WHO launched a global effort to eliminate industrially produced trans fat by 2023. And here the trans fats or the trans fatty acids are a form of unsaturated fats, and they come in both natural and artificial forms. And the artificial trans fatty acids are formed when hydrogen is made to react with the oil to produce fats resembling pure butter or pure ghee. So here we have the saturated and unsaturated fats. So the saturated fats are the butter, ghee, cheese, etc. And the unsaturated fats, that is the dry fruits, walnuts and others. And the trans fats are your cakes, breads and pancakes. So all these junk foods. And here, what is actually a saturated fat? So that means here in the chain, so there is no double bond at all. So all are single bonds. So all carbon atoms are saturated with the 4 bonds. But unsaturated fatty acids, so they have double bonds like this. And here we have the cis fatty acid and trans fatty acids. So among the unsaturated fatty acids, if there is a double bond and the hydrogen atoms are attached to only one side, so it is called the cis. And if it is attached to both the sides, it is called the trans fatty acids. So if we take up a cis fatty acid and we react with the hydrogen, so here this hydrogen, it will be shifted to this side, and the structural formula changes, and thereby the trans fats are being born. And here the major sources of the artificial trans fatty acids are the partially hydrogenated the vegetable oil or the vanaspati ghee or the margarine. So this is the best example for a trans fat, and it poses a high risk of heart disease than saturated fats. So this is because saturated fats, they raise the total cholesterol level, and trans fatty acids, they not only raise the total cholesterol level but also reduce the HDL. So friends, here we have 2 lipoproteins. One is the high density lipoprotein, that is the HDL, and the low density lipoprotein, that is the LDL. So this LDL is unwanted fat. So this our body also produces. But this HDL is a wanted fat, it is a good fat. So we require more and more HDL and less of LDL. But these trans fatty acids, they will reduce this HDL. So that's why they become double dangers. So these are some of the facts. Then come to next, how this partial hydrogenation happens. So that we have here. So 2 radicals will come and attach here. And as and when the radicals move out, this hydrogen will come and attach to the other side. So this is how the trans fats are being formed. Then come to next, the food fortification. So here, as per the Food Safety and Standards Authority of India, so it says that 1 kg of fortified rice, it should consist of iron, folic acid, vitamin B12, and then micronutrients like the zinc, vitamin A, B1, B2, B3, etc. So which should be added to what extent is not the matter of discussion here. So here food fortification is, it is just mixing the rice with some nutrients. So here, neither hybridization occurs nor the genetical modification occurs. So here I am not editing any gene. At the same time, I am not adding this at the time of growth of the plant. So nothing. So I am taking the normal rice. So in that normal rice, I am mixing various nutrients so that this fortified rice, it is a mixture and it is not produced by the rice per se. So remember the difference between the hybridization, which means that I am growing these nutrients from its birth, and genetical modification, I am modifying the genes, and food fortification, just mixing of some nutrients. Then come to next, the norovirus. So recently a highly contagious virus named the norovirus has been detected in Kerala, and it is a group of viruses that leads to gastrointestinal illness. So it is basically the diarrhea, dysentery and others are the symptoms, and it causes inflammation of the lining of stomach and intestine, and in addition to severe vomiting and diarrhea. And norovirus is resistant to many disinfectants and can heat up to 60 degrees Celsius. So that means merely steaming of food or chlorinating water does not kill the virus. So they are highly chemical and heat tolerant. Then the virus can also survive many common hand sanitizers. So even sanitizers can't kill these noroviruses. Then a person can be infected with different varieties of norovirus several times in their lives, but developing immunity to one type doesn't provide protection against the other varieties. So it is very similar to common cold here. So just because I am immune to one type of cold, but if I go to other place and if I drink the water of that place, I can get infected again by the common cold virus of other variants. Then the virus can be transmitted through contaminated surfaces or the contaminated food. Then the virus can primarily be transmitted from the oral fecal routes. So that means by the saliva, by the feces and others. Then diarrhea, vomiting, then stomachache, then gastrointestinal problems are the common symptoms. Then, while there is no specific treatment available to cure the virus, the generic medication, that is the diarrhea, vomiting, can be curbed with the help of the dysentery related medications. So these are few facts about norovirus. Then come to the last part. Friends, as the common saying goes, that is the pain is gain. So here, this applies to the every field for that matter. So for example, here also, it requires some efforts for you to learn something. If you see a concept, so you have to analyze it from various perspectives. You have to read it again and again to memorize, and only then you will learn a concept. So a person who has the mindset to incur the pain, and only those people who are willing to persevere, so they can succeed. So make sure that your brain is much stronger and it perceives everything easily, so that the task in front of you, it remains to be feasible, and your brain, so that overcomes so whatever difficulties that you face. So make sure that the strongest brain, so that will achieve the greatest things. So do it. All the very best. Good luck, friends.